Hi, we're here at the Cold Spring Harbor Genome Integrity and Stability meeting. I'm Krista Bledsoe, and I'm an editor at Molecular Cell, and I'm here with Wei Yangs, who has a lab at the NIH. Thanks so much for being here with us today.
Thank you. It's great being with you.
Yeah, could you tell us a little bit about what your lab works on?
Yeah, my lab works on a lot of things coupled with repair, recombination, and replication. And I started interested in DNA when I was a graduate student, and Fred Ott was our professor, and I was at Columbia, and that was like the mid-1980s, and they just discovered, gene VDJ recombination, which is essential for making antibody and antigen receptors, and I just was fascinated. It took a long way for me to get to the point I can do structure and elucidate.
Yeah.
Yeah, like one like two recombinase.
Interesting.
And then just expanded to recombination when I was postdoc with Tom Stites. And then my colleague, Peggy C., introduced me to mismatch repair. My other colleague, Roger Woodgate, enticed me to work on translesion synthesis, and the one step by another step, everything leads to replication fork, or many things leads to replication fork.
Yeah, interesting. Yeah, you gave a great talk earlier this week, and you mentioned that you think the last frontier of the central dogma is replication. So can you tell us a little bit about what you think those open questions are still in replication?
Right. So it's when I was a graduate student, three things: repair, recombination, replication, three entity. But actually, repair and recombination become only critical, most critical, coupled with replication. And so the question is how they are coordinated.
Yeah.
And there are a lot of things, whether it happened, like repair. In a lunch we were talking with David Cortez, and the protein he discovers basically cell want to prevent base excision repair. Once it's an A basic, leads to Nick, and Nick's double strand breaking replication. But somehow APE1, which make the Nick, is actually quite well expressed during S phase. So, all those questions, what cell try to do, and how they coordinate different things for one goal to finish replication, and how they interact. Every talk, every day we heard how things are added, right? Yeah. One more protein to coordinate.
Yeah.
Yeah, that's absolutely unknown. Not just like structurally how to define it. There's still many players out there.
Yeah, interesting. Yeah, and you gave a great talk about the structure of the replosome, and you mentioned the trombone model that's out there, and sort of data that you have that conflicts with that. Can you tell us a little bit about what was in your abstract and how?
Yeah, so trombone model is aesthetically pleasing because DNA 2 template strand, parental strand are anti-parallel. So human really like symmetry. Truly like symmetry. We think human is beautiful because they did survey. And so to make two synthesis strand in opposite direction, it looks awful. So trombone model is very, very pleasing, and that replication fork is, in my mind, is always a Y shape.
Yeah.
But then nature didn't obey. So when we found it's not like a polymerase following helicase, and we scratch our head, and when we have the structure showing, visualizing how the two strands are separated, then it makes sense that because helicase need to pull the DNA strand apart, the best way pulling is perpendicular to the helical axis, and it only pulls one strand, so it needs a helper to on the other strand, not particularly pulling, even just fix it is a lot easier to separate. So that's a unit. So the polymerase, whoever work with leading strand or lagging strand with helicase is a unit. And their orientation is only important right junction there, 90 degree to the parental. But once it's 2 steps away, it doesn't matter anymore. And so there's a lot of freedom for the lagging strand, how they coordinate it. And nature, all they want is make the product and make it error-free. It's not really pleasing our eyes.
Yeah. What are the next steps for your lab on that project?
So, of course, obviously, is to get the mammalian reprosome. Yeah, it's obvious. But there are a lot of people much better qualified or prepared. So I think, it's really a question when one can do things actually uniquely contribute to the field, and when one, couldn't. So we have still, looking for the right angle.
Yeah, that makes sense.
Yeah.
Yeah, we were chatting earlier about how structural biology's changed and cryo-EM kind of lowered the barrier to entry, and AlphaFold has changed things.
Yeah.
Has that changed the way that you work in your lab or the kinds of projects you take?
Yes, it definitely. It hasn't changed how the project we take us. Maybe 15 years ago, it's an interesting process. In the early days, structural biologists couldn't do big one, crystallography, NMR, all small pieces. So when we're facing a big chunk of protein, what we do is divide, conquer. But even by 2000, it was very dissatisfying, because when you take out a little piece out, you have the structure, you have everything, but it doesn't mean that's how it works. And even by 2000, little after 2005, my lab already changed the orientation to get bigger, complex, as big as possible, but complete, not as big as complete as possible. So 2015 is the transition time we start to have cryo-EM. A lot of projects that's hold up couldn't move forward, and now have a chance to move forward. So cryo-EM really, give us lifeline. And in terms of AlphaFold, is very useful for us, is because some of the part of structure we have usually big complex. Some part of the structures are no, and AlphaFold give us the best starting model. But the thing is, I have structure we solved back in 2000. I presented here. It's one state of protein DNA complex. We were missing the most critical state, which is ATP bound, mutase binds to mismatched DNA. It was hypothesis there. You just never see it. So AlphaFold couldn't predict what's the second state and third state.
Yeah. Interesting.
Yeah.
Yeah, that's really neat. Thanks so much. This was a really nice conversation. It was great to hear more about your work.
Thank you, Krista.
Thanks.
It's really nice talking with you.
You as
And
well.
we spend a lovely time here through the week.
In person at the meeting.
Yeah.
Thanks.